Let me just get this going. So thank you also to the organisers for giving me the opportunity to come and letting me kick this session off. And what I would like to do is give a very sort of top level presentation describing a little bit about the things you need to think about before you even embark on doing a time-resolved experiment. And then we've got 4 other speakers in this session who are really sort of living the dream and working at the coalface of these experiments, who are going to show you some much more details of the kinds of things that are possible, and also show you some really exciting results that I'm also excited to see. So why might you want to do a time-resolved experiment? And I wanted to give a special thank you to Steve Munch yesterday for giving a really good introduction to time-resolved experiments, and I enjoyed the cat example very much. And really we embark on these kind of experiments because what we want to do is observe a system as it functions, and that gets us sort of deep into in my view the raison d'etre of structural biology, which is to try and understand the relationship between a macromolecular structure, its function, but also its dynamics. So how do these fabulous molecular machines work? And how do they work really with mechanistic detail? That's really what gets me going in the morning. So questions you might want to answer, or that you could answer with time-resolved experiments, for example, are how our relatively slow macromolecular dynamics connect to extremely fast chemical steps. And this is something that's fascinated me for years, how femtosecond chemical reactions can be controlled by proteins, where the protein dynamics are many orders of magnitude slower. So how do these dynamics interplay over these huge orders in time? You could also ask questions like, how are signals communicated across macromolecules? How does a ligand binding on one side of a molecule somehow trigger an effect on the far side? How is this information transmitted? And of course, you can also start to get into the questions about how small changes, how simply changing a single amino acid can have a huge functional effect. And all of these types of questions are questions you can answer with time-resolved structural biology. So what sort of timescales might we be interested in? And I've sort of focused on the things I'm interested in. You might be interested in things that are much slower. So you've got time across here. We're going from chemistry steps that happen on the femtosecond. And then we've got seconds, which is sort of where I start to lose interest in things that are much slower than seconds. Some of you might be. But within this, there's an awful lot of stuff going on. So we have our allosteric transitions that happen somewhere between microseconds and seconds, depending on how much has to move. Enzyme catalysis also lives in this regime, where the rate determining steps of enzyme catalysis are also mostly in that hundreds of microsecond, millisecond area. But these are coupled to much faster motions, so side chain rearrangements, water structure rearrangements, helix coil transitions, loop hinge dynamics, and then all of these couple down into chemistry. And if you're interested in mechanism, you actually have a huge number of tools available to you, okay? And I've highlighted these here because I think the message, if you remember nothing else about my talk, is that much as it pains me to say this at a study weekend meeting, a crystallographic experiment might not be the right one for you, okay? And so spectroscopic tools let you get at either very detailed local structural information or global dynamic information over this whole time range. And sometimes a spectroscopic experiment could be exactly what you want to do. You've also got the magnetic resonance type spectroscopies, and we've heard some nice talks about NMR, which also gets us exactly at these timescales, but often looking more equilibrium behavior rather than driven behavior. Steve introduced us yesterday to single particle cryo-EM and the progression coming there to move into time-resolved studies, which is super exciting. And then, of course, we've got our scattering and diffraction type experiments, so small angle, wide angle X-ray scattering, but also diffraction, and those are both at synchrotrons and CFELs. And I've split these two out because the experiments are slightly different beasts. The CFELs really allow us to come down into these chemical time regimes really down to femtoseconds, but people are doing experiments with much slower timescales at CFELs very successfully. Synchrotrons are sort of pushing down. So 100 picoseconds is a feasible experiment to synchrotron, if that's a question you want to answer. Most of the work we're doing at the moment is sat in this millisecond area at synchrotrons with diffraction. So if you're thinking this is the kind of thing you might want to do, you might be wondering, where do you start? Okay, and my first piece of advice for you is to think extremely carefully about what the scientific question is you actually want to answer, okay? Because this is going to define the kind of experiment you should do. So you really need to be thinking about what kind of structural information do you need? Do you just need information about what's going on around an active site? Do you need information about a global structural rearrangement? Do you want to see everything? What do you need to know to answer your science question? Also, what kind of time resolution do you need? And this is linked to the question of what kind of time scale you need to access. You might need to access a species that has a very short lifetime, but takes many minutes to form. And that means you need to think about doing a different kind of experiment to if you want to look at something that maybe has a short lifetime, but forms very quickly. And considering these things very carefully up front will help you do the right experiment for you. You also need to think about experimental limitations. Are there limitations on the amount of sample you can make, for example? And this again might constrain your sort of experimental freedom in the types of things you can do. And finally, you need to think about how you synchronize the system you want to look at. And you'll hear more about this from some of the other speakers. But why is this synchronization important? It's important because the majority of the experiments we do are ensemble measurements, particularly if we're wanting to look at structural information. People are pushing, trying to do single particle, genuine single particle experiments structurally. I think that's still quite a ways off. And really what we're doing usually is an ensemble. And that means we see all the conformations of our macromolecule that are present in the sample we're probing. And this, of course, are the frames. Clearly, the cat will be the new hot example, but the historical example for time-resolved crystallography are horses. And these are freeze frames from Muybridge's fabulous time-resolved experiment trying to understand whether a horse has all its feet off the ground at the same time when it's galloping. But in an ensemble measurement, we would see all of these conformations at once, which means we see this sort of superimposed, smeared out state where our ensemble structure that we observe is averaged over space and over time. And normally this means we lose dynamic information, we often lose structural resolution, and crystallographers, you know that these flexible regions in your structures are often the bit you're most interested in and can be really the bits that are hardest to see. So how do we go about actually doing these experiments? And the simplest approach to getting at this kind of information is one of sort of stop motion of trapping. And I would say here, my second message for this talk is don't make your life harder than it needs to be. All right, if you can answer your question with a relatively simple experiment, do that. You will be much happier. Supervisors, your students will be happier, and you'll get your results more quickly. So trapping experiments, these are oldies but goodies, okay? So what do we do in such an experiment? We set the experiment up so the reaction stops at a certain point, all right? And the advantage of this is that it doesn't matter what the time scale or the lifetime of a species is. If the experiment can't progress beyond this point, that's the state you're going to see in your sample. So we've basically synchronized by letting the whole system pile up at a certain state. And these kind of tools are extremely well established and have been used in enzymology since enzymology started, and in structural enzymology since the 60s. And what I call mechanistic trapping, this real arresting of the reaction, can be used regardless of the rate of the reaction steps that you're trying to look at, as long as you can come up with a clever way to trap it. So this could be altering your reaction conditions so the system can't turn over. Maybe you make a mutation so it can't turn over. Maybe you alter the substrate, or maybe you simply accumulate your steady state species. And an example here is one I worked on now a long time ago with Carrie Wilmot at the University of Minnesota, and I'm just showing you the business end of this enzyme. There's a green colored cofactor, it has 2 oxygens on, and this thing binds methylamine, turns it into formaldehyde, and then goes through a series of reoxidation steps to bring us back to the resting state. And we can design a mechanistic trapping experiment for this relatively simply. If we don't give it any substrate, we get the resting state. If I give it substrate but no electron acceptor, I get this reduced state. If I give it enough electron acceptor to take 1 electron off but not a second, I get my semiquinone state. And so these kind of trapping experiments let us really nicely create the state we're interested in, and we can interrogate it at our leisure, which is fabulous, which is why this is totally the way to go if you can do it. You can also think about doing cryo trapping or freeze quench style reactions, and these are the types of experiments that Steve was showing you yesterday, where you let your reaction run, but the reaction itself is slow enough that you can trap this by rapidly cooling it. All right, so you freeze out the state you're interested in. If your reaction's a little bit too fast for you to do this, you can actually run the reaction itself at a lower temperature to reduce the reaction rates. And if you're interested in this kind of stuff, I can highly recommend this book by Pierre Dezou called Cryobiochemistry, which actually goes into how to do lots and lots of enzymology reactions below 0 Celsius, so down at sort of -10 or -20 degrees, where you can really slow things down and slow intermediates down to the point where you can look at them. But most of you probably aren't here for trapping experiments. You're here because trapping experiments haven't answered your question. And so what do you do? And this could be that you can't trap the state you're interested in. Maybe there's no obvious way to mechanistically trap it. Maybe it forms too fast, or it's too short-lived for you to cryo trap it. Or maybe you want to know really about the transitions between these metastable states that we trap out in a trapping experiment. And in this case, you do need to do an experiment that's time-resolved. Now, all time-resolved experiments essentially follow the same scheme. We have some kind of trigger, we wait a period of time, and then we probe, okay? And then at some point the detector is read out. And this scheme is known as pump probe, was first proposed by Porter. And what determines the time resolution that you can achieve in these experiments is whichever is slowest, all right, of your pump pulse width, so how quickly you can initiate your reaction, this time delay, and the width of your probe pulse. So how long do you need to acquire data to have something usable when you read your detector out, okay? So whichever of these 3 things is slowest determines the time resolution that you can achieve in your experiment. However, I'll put a caveat on this, that's really all about signal to noise, okay? So what we want is a nice system where we get lots of signal, we have limited background, or as little background as we can get away with. And this is actually really, really important. And so why should you be worrying about signal to noise? So in any time-resolved experiment, we get a series of data sets recorded at different time points, and I'm using here crystallographic nomenclature, but this would be the same for whatever, whether you're doing spectroscopy or scattering. So we have some kind of structure factors at a time t, or a series of time t's, and we have a dark structure, which we recorded at time 0. And in time-resolved crystallography, we then go on to calculate difference electron density maps, and these are our delta F, our changed structure factor at a time point, is therefore our structure factor at that time point minus the dark state, and we use the phases from the dark structure, so we're looking for these changes. And what's important for you to remember here is that there's always some heterogeneity in these data, and this is reducing the amount of signal you have. So this can come from fractional excitation, and you'll hear a bit about this in the talks that come later. And it can also be because you get mixtures of different intermediate states. And this figure here I've swiped from a nice review by Maria Schmidt. As your reaction progresses, our starting material might be disappearing and intermediates coming up. But if we look at any of these time points, what you see is we're seeing mixtures because our ensemble is reacting in a slightly stochastic way. And if you've looked at single molecule enzyme kinetics, we know that individual molecules can be extremely variable in when they trigger. And when we're looking in an ensemble sample, all our molecules are reacting at slightly different times, even though the macroscopic rates of these steps might seem very, very clear cut. And this means that our structure factor at time t is actually a mixture of unreacted sample, but also the contributions from the different amounts of intermediates that are present at the different time points. And that all of that is taking your signal down for the things you're actually interested in. So when you collect time resolved data, you have to collect enough data to be sure that you can really see the differences. And the lower the fraction of your sample that reacts in the beginning, the more data you need to be sure that you've got something there. And I want to say here, a complete data set is usually not sufficient. So just sampling all your reflections once doesn't give you enough signal to noise to be really sure that you're seeing these changes. So this is something you have to think about, and it's much like whether your ligand is there or not, whether your time resolved intermediate is there or not. These are things you need to think about. So how have these time resolved experiments, sorry, I missed a slide. How do we increase the signal? This is dependent on both source and sample. So we can make brighter beams. This is Darwin's law, by the way, which tells you that the intensity in a single reflection is related in this way to the intensity of the incident beam, to the volume of your sample, the volume of your unit cell. We're inversely related to that, and also to the scattering power of your sample. And these two things are probably things you can't do much about without changing your either crystal conditions and your crystal habit. You're certainly unlikely to be able to do much about the scattering power of your sample. But of course, you could make your X-rays brighter, you could make your crystals bigger, and this increases your signal. And historically, this is how things have been done. Just a reminder here that the intensity that's coming from your time-dependent state, again, is related to how much of your sample has reacted and to how many states your sample is in. So historically, these reactions or these types of experiments have been done on large single crystals, okay, to have as much scattering material in the beam as possible to get nice, strong data. And they've used Lowy radiation, so polychromatic X-rays, to maximize both the per shot information and the time resolution that can be achieved. And for fast experiments, and by fast I'm talking sort of sub-millisecond experiments, they really needed state reversible systems. And what I mean by state reversible system is one where once the reaction has happened, the system relaxes back to its starting state. So you can then turn your big crystal and redo the experiment. So we build up our complete picture of reciprocal space. Most of biology isn't state reversible, so this was, of course, problematic. If you don't have a state reversible sample, you needed lots of these big crystals, which made these experiments extremely difficult for anything that was really, really biologically interesting. Now, serial crystallography, 10 years old now, 2011 was the first Henry's groundbreaking paper. This changed the game. All right, into how we think about doing these kinds of experiments, because they address the challenge both of reaction, they address this challenge of reaction irreversibility. And in our serial experiments, we take each shot from a new sample. So I'm sure you're all aware of this now, whether we do lots of flavors. You can do mesh and collect, where we have many crystals on a grid and we scan across the grid. You can think about doing helical and grid scans. At synchrotrons, we call this serial synchrotron crystallography. At XFELs, it's serial femtosecond crystallography, which was the subject of Henry's original paper. But because we're taking each shot from a new sample, that means we don't care that the reaction was irreversible, because we're able to put a new crystal into the beam each time and start our experiment again with this new sample. Now, depending on how you're doing your experiments are how you're delivering your sample and your sample availability, that already puts some practical limits on the sample size that you can use, and you'll hear some more about that again later on in this session. But also, we have some sample constraints that are associated with how you're initiating your reaction. And I want to remind you that crystallography is determining the average structure of the ensemble of molecules in the crystal. And that means you need all the molecules doing the same thing as much as possible. So with all our caveats about fractional excitation and only a small portion of the molecules reacting. So if we're not trapping, that means we have to somehow synchronize the reaction, and that means we somehow need a way to say, start now, all right, go. This is your point to trigger. And there are two basic ways we can think about doing this. We can do this by mixing, by rapid mixing, and we can do this using light. And these two ways to initiate a reaction are associated with two concepts that link back to how we define our sample size. So one of those is critical depth, okay, so how thick my sample is, and the other is laser penetration. And again, you're going to hear about more of these later, but I just want to quickly put these in context. So critical depth is the maximum distance a ligand has to diffuse, okay, so if you're adding in a mixing experiment, adding your ligand, so the maximum distance the ligand can diffuse for the diffusion rate to be faster than the thing you're trying to look at. All right, so if the diffusion rate is slow compared to what you're trying to look at, you're never going to get a clean initiation and a clean, or as clean as possible, electron density. And depending on the reaction rate of the species you're looking at and the buffer conditions, this can be really, really very variable. This is pretty much real time. This is a large crystal, a few hundred microns, and you saw this is actually the system we looked at the beginning. I've added some ligand to the buffer around this crystal, and you're seeing it diffusing in and the system reacting. And so this can take a period of time. Again, this is something that people have looked at a lot, and there's a fabulous review from Mackinnon and Fink from 1977 where they looked at the reactions of enzymes in crystalline solution states. And the parameter I want you to look at in this table is this lambda c, which is the critical depth in micrometers. And for these enzymes, this is the thickness of crystal that you could have in order for the diffusion rate to be faster than the reaction step they were investigating. And you can see that for some of these enzymes, this is relatively thick. So this is the distance from the outside of the crystal to the center of the crystal, okay? So sort of 6 microns looks reasonable, but we have some very fast reacting systems where we're down, for example, at 100 nanometers. And so we're starting to get to very, very short diffusion lengths for very fast reactions. And so this is why mixing is not suitable for super, super fast reactions that you might want to be interested in. We also have our issue of laser penetration if we're activating by light, and I've got here my laser beam and my crystal. And of course, the reason your laser works to activate your sample is because it's interacting, the laser photons are interacting with the chromophores in your sample and driving light-dependent reactions that are hopefully triggering the system you're interested in. And how much of your laser gets through your crystal and how far your laser can get through your crystal is related to the absorbance of your system. So if you have a highly absorbing crystal, you'll excite the molecules at the front of the crystal, but the molecules at the back of the crystal won't be reacting. But because our X-rays are much more penetrating, your X-rays will be measuring data from this whole volume, all right? And that means that you're basically including unreacted material in your data set that's going to contribute back to that reduction in the signal to noise for the species you're really interested in. So we can easily measure the absorbance, and that you can easily work out what kind of laser powers you need. Be aware that if you put too much laser power in, you start to get weird nonlinear effects where the laser itself is causing changes in your sample that are maybe not the physiological ones you're interested in. You can minimize this a little bit by coming off the maximum absorbance, but still getting reasonable excitation. This can also, will of course increase your laser penetration. But as a rule of thumb, for both mixing and laser penetration, well, certainly for lasers, for mixing it really depends on how fast you want to go, but certainly for laser activation, a sample thickness of about 10 micrometers should be okay. All right, and I say should be okay for most of, unless you've got some crazy octaheme monster. So where can you do these experiments? And I would argue now that time-resolved MX in its current state really requires serial crystallography. You can do these experiments with large single crystals, but you're setting yourself up for a miserable time. So if you can do it by serial, please use serial. There's a huge range of sample delivery platforms, and you'll see examples of many of these in the talks in the rest of the session. And they include capillaries, microfluidics, jets, solid chips, tape drives, and there's all kinds of cool ideas that are still coming out as the best way to do these experiments. You can do these experiments at all CFELs. All CFELs offer the possibility for these time-resolved experiments. And these are really your only option for time resolutions better than 100 picoseconds. Okay. You can also do these experiments at synchrotrons for the slower time resolutions, but you do need non-standard end station setup. So you can't just walk up to your classical MX beamline and announce that you've arrived to do a time-resolved experiment. There are an increasing number of dedicated synchrotron end stations for time-resolved MX, and I'm bound to have forgotten somebody, so apologies if I have. There's TRex at Petra 3, Biocars at the APS. ID29 is the new end station that Daniele is developing at ESRF, but of course ID9 has long been a leader in time-resolved crystallography. I put it in brackets because you can also do non-crystallographic experiments on ID9. I24 at Diamond Light Source is beautifully set up for time-resolved experiments, and there are lots of other end stations that are capable of SSX and in principle capable of time-resolved SSX, but where the end station isn't dedicated to that. So you have to go and do some modifications, maybe installing a laser, maybe installing a new sample holder or sample environment in order to do your experiment. But you will see some examples, I think, in some of the talks that are to come of work that's been done on more classical end stations, but where they've been modified for that experiment. So that's bringing me to the end of my presentation, and I just wanted to highlight, if you are interested in these things, obviously my favorite beamline, I'm sorry Robin if you're listening, is now TRex at beamline P14 at Petra 3, where we've built a dedicated end station for time-resolved crystallography, and this has been operational since March 2019. We can offer you initiation by mixing or by lasers. We've got nice live hit rate feedback. So this is a scan of a solid target, and every blue spot there was a crystal, and with automatic data processing. So we try and hold your hand through the whole experiment so we can send you away with process data that you can then do something with, as opposed to just giving you a whole load of images and wishing you good luck. So I shall stop there, and hopefully there's a little bit of time for questions, and if not, we can all catch up afterwards.
Thanks very much. Great introduction. There are a couple of questions. We don't have too much time. And Franz first outed himself as also a key